Good day, grade 8 ST students. I hope this video lesson find you in a safe and healthy condition amidst this pandemic. This is Mr. Marin Rodrigo D. Marinas III, your teacher facilitator for this asynchronous online class in biotechnology. Before you continue learning our topic, please be reminded of the following. Number 1. Choose a study space conducive for online class. Number 2. Set a time for you to finish all the prescribed activities given by your teacher. Number 3. Take notes while watching the video lesson. Number 4. Listen carefully to the instructions given by your teacher. And number 5. Reach out to your teacher with questions relevant to the topic of the video lesson. Since this is an asynchronous online class, your attendance will not be checked on schedule. Your submitted output will serve as your attendance, so make sure to submit all your outputs on the given schedule. In addition to those reminders and instructions, you must have the hard copy of the teacher-made activity sheet provided to you a week before the uploading of this video lesson. Browse the activity sheet and check for missing pages. Read the instructions carefully and reach out to your teacher for some clarifications. Before we have our lesson, let's have first activity number 1 entitled Break the Chain. For the directions, identify the components of the chain of infection being described by the following statement. Choose your answer from the box below and write the capital letter of your answer on the space provided before each number. For statement number 1, it refers to the habitat in which the agent normally lives, grows, and multiplies. This may include humans, animals, and their environment. And the choices are A. Host, B. Mode of transmission, C. Portal of exit, D. Infectious agents, E. Portal of entry, and F. Reservoir. For statement number 2, it is the path by which a pathogen leaves its host. It usually corresponds to the site where the pathogen is localized. For statement number 3, this refers to the methods on which an infectious agent is transferred from the reservoir to the host or from host to another host. Let's have statement number 4. These are disease-causing organisms or substances such as bacteria, fungi, and viruses. And lastly, statement number 5, it is the manner in which a pathogen enters a susceptible host. This must provide an access to tissues in which the pathogen can multiply or a toxin can act. Before proceeding to the next part of this video lesson, make sure that you are through with answering this previous activity. If you're done, you may now continue playing the video lesson. If not, you can still continue doing this activity before proceeding. Let us now have your activity number 2 entitled Do You Still Remember? Look at the photo montage presented on the slide. Do you still remember the last time you had your vaccine? If yes, please write a 5-sentence paragraph about your experience. If you can't remember, just write a 5-sentence paragraph about your thoughts about vaccination. Before proceeding to the next part of this video lesson, make sure that you are through with answering this previous activity. If you're done, you may now continue playing the video lesson. If not, you can still continue doing this activity before proceeding. Let's now continue with activity number 3 entitled List Me Down. Flash on your screen is the word vaccination. Your task is to list down 4 words that are related or relevant to the word vaccination. Before proceeding to the next part of this video lesson, make sure that you are through with answering this previous activity. If you're done, you may now continue playing the video lesson. If not, you can still continue doing this activity before proceeding. For a pathogen such as bacterium, fungus, virus, or other disease-causing agent, our body's internal environment is a nearly ideal habitat. Our body offers a ready source of nutrients, a protected setting, and a means of transport to a new environment. From the perspective of a cold or flu virus, we are a wonderful host, but from our vantage point, the situation is not so ideal. Fortunately, adaptations have arisen over the course of evolution that protects ourselves against many pathogens. In this case, dedicated immune cells in our body fluids and tissues specifically interact with and destroy pathogens. Together, this makes our body's immune system. This system enables our body to avoid or limit many infections. These immune cells produce receptor molecules called antibodies that bind specifically to molecules from foreign cells or viruses and activate our defense responses. The specific binding of immune receptors to foreign molecules is a type of molecular recognition and serves as the basis for the two types of immune defense found in our body, the innate immunity and adaptive immunity. Before we proceed to the next part of our discussion, please answer this question using 1 to 3 sentences only. The question is, how does our immune cells help in defending our body against pathogens? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of the video lesson. In innate immunity, the first lines of defense prevent pathogens from gaining entrance to our body. This may include barrier defenses such as our skin, mucous membranes, and secretions that lines the entrances and exits of our body. Innate immunity is a rapid response that relies on a small set of receptor proteins that bind to molecules or structures that are absent from our body but common to a group of viruses, bacteria, or other microbes. Binding of an innate immune receptor to a foreign molecule activates internal defenses, enabling responses to a very broad range of pathogens. If a pathogen breaches these barrier defenses and enters our body, our immune system must detect these foreign particles and distinguishes it from the rest of our body cells. This internal defense system may include phagocytic cells, natural killer cells, antimicrobial proteins, and inflammatory response. Meanwhile, in adaptive immunity, molecular recognition relies on receptors, each of which recognizes an antigen, a feature typically found only on a particular part of a particular molecule in a particular pathogen. As a result, recognition and response in adaptive immunity occurs with tremendous specificity. The adaptive immune response, also known as acquired immune response, is activated after the innate immune response and develops more slowly. The names adaptive and acquired reflects the fact that this immune response is enhanced by previous exposure to the infecting pathogen. Therefore, there are several ways in which we can enhance this type of immunity. Before we proceed to the next part of our discussion, please answer this question using 1 to 3 sentences only. The question is, what is the main difference between innate and adaptive immunity? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of this video lesson. As mentioned earlier, there are several ways in which we can enhance our adaptive immunity. This can be through passive natural immunity, passive artificial immunity, active natural immunity, and active artificial immunity. The terms natural and artificial refers to the method of exposure. Natural exposure implies that contact with the antigen occurs as part of everyday living and it is not deliberate. On the other hand, artificial exposure is the deliberate introduction of an antigen or an antibody into our body. Meanwhile, active immunity results when an individual is exposed to an antigen, either naturally or artificially, and the response of the individual's immune system is the cause of the immunity. On the other hand, passive immunity occurs when another person or an animal develops immunity, and the immunity is transferred to a non-immune individual. Let's go over each type of adaptive immunity. For passive natural immunity, it is achieved when antibodies from the mother are transferred to her child across the placenta or in breast milk. For passive artificial immunity, it is attained through the injection of antiserum to a non-immune individual. Antiserum are antibodies that are found in the serum of the blood plasma without the clotting factors. This may be produced by another person or an animal. Antisera are now available against microorganisms that causes diseases such as rabies, hepatitis, and measles, and for bacterial toxins such as tetanus, diphtheria, and botulism, and venoms from poisonous snakes and spiders. Meanwhile, active natural immunity is achieved when antigens are introduced to an individual through natural exposure. Because the individual is not immune during the first exposure, he usually develops the symptoms of the disease. Active artificial immunity is attained when antigen is deliberately introduced to an individual to stimulate the immune system through the process of vaccination. Injection of the vaccine is the usual mode of administration for this type of adaptive immunity. Before we proceed to the next part of our discussion, please answer this question using 1 to 3 sentences only. The question is, what are the main differences between the 4 ways of achieving adaptive immunity? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of the video lesson. Let us have again an activity. This activity is called immunoprofile. Here is what you're going to do. Right now, you can see a list of words that are related to the different types of acquired immunity. Your task is to place those words into their proper groupings. There will be some words that can be used more than once. The words are antiserum, deliberate, natural exposure, placenta, weakened microbes, antivenom, milk, not deliberate, vaccination with symptoms. Shown to you right now is the table in which you're going to place the different words. The first column is passive natural, followed by passive artificial, the third is active natural, and the last is active artificial. Before proceeding to the next part of this video lesson, make sure that you are through with answering this previous activity. If you're done, you may now continue playing the video lesson. If not, you can still continue doing this activity before proceeding. At this point, we are going to focus now with vaccination. So let us start our discussion with its early history. On May 4, 1796, a young British physician named Edward Jenner embarked on an experiment to find a way to protect people from smallpox. Smallpox epidemics were greatly feared. This disease periodically swept through Europe, killing thousands of people and even wiped out 90% of Native Americans when Europeans first settled in the Americas. Smallpox is caused by an orthopox virus known as variola virus. Transmitted by the respiratory route, the virus infects many internal organs before their eventual movement into the bloodstream. That leads to the infection of the skin and the production of a more recognizable symptoms such as lesions on the epidermal layers of our skin. Jenner's experiment started when a young milkmaid informed Jenner that she couldn't get smallpox because she already had been sick from cowpox, a much milder disease. So Jenner put the girl's story to the test. First, Jenner collected scrapings from cowpox blister. Then he inoculated a healthy 8-year-old volunteer, scratching the person's arm with a cowpox-contaminated needle. The scratch turned into a raised bump. In a few days, the volunteer became mildly sick, but recovered and never again contracted either cowpox or smallpox. The process was called vaccination from the Latin word vacca meaning cow. Louis Pasteur gave the process this name in honor of Jenner's work. Pasteur then called the material vaccine for cultures of a virulent microorganism used for preventive inoculation. Before we proceed to the next part of our discussion, please answer this question using 1 to 3 sentences only. The question is, how significant is Jenner's experiment regarding vaccination to us humans? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of this video lesson. Vaccines are suspensions of organisms or fraction of organism that is used to induce immunity. As mentioned earlier, the term vaccine was coined by Pasteur as recognition for the monumental work of Jenner in battling smallpox during the mid-18th century. Vaccination is an important program to develop herd immunity among the population. Herd immunity is described as the phenomenon where outbreaks are limited to sporadic cases because there are not enough susceptible individuals to support the spread of the epidemic. Around 80% of a population is usually the ideal number for herd immunity to be achieved against certain diseases. There are now several types of vaccine. Some of the newest vaccines take full advantage of knowledge and technology developed in recent years. In this video lesson, we are going to use the context of COVID-19 pandemic to discuss the different types of vaccine produced by various companies, such as live attenuated vaccine, inactivated vaccine, subunit vaccines, viral vector vaccine, nucleic acid vaccine, and toxoids. Let us start with live attenuated vaccine. This type of vaccine uses living but attenuated or weakened microbes. This type of vaccines more closely mimic an actual infection as it stimulates a robust immune response without causing serious diseases. Live attenuated vaccine may not be safe for those with compromised immune system. Live attenuated vaccines are since used to combat MMR or measles, mumps, and rubella, as well as chickenpox. Currently, Codagenics of the USA and Indian Immunological Limited is currently producing this type of vaccine for COVID-19. Meanwhile, inactivated vaccines uses the whole virus after it had been killed with heat or chemicals such as formalin or phenol. It is generally safe because the virus is already dead and it is easier to produce compared to other vaccine. However, it is not as effective as the live attenuated vaccine, so there is a need to regularly check the quality of this type of vaccine in clinical trials. Inactivated vaccines used in humans include those against rabies, influenza, and polio. On the other hand, COVID-19 vaccines developed by the Chinese companies Sinovac and Sinopharm, as well as Russia's Gamaleya, which developed Sputnik V, are classified as inactivated vaccines. Subunit vaccines uses the antigenic fragments of the microorganisms such as viral surface proteins to focus our immune system to a single target. Unfortunately, this type of vaccines may not stimulate a stronger immune response compared to the other types of vaccine, so there is a need for chemical additives to boost a long-term immunity. Subunit vaccines used in humans include those against pertussis, hepatitis B, and human papillomavirus or HPV. In the case of COVID-19, Novavax of USA and AdaptVac produce this type of vaccine. Viral vector vaccines take a harmless virus and uses it to deliver viral genes to build immunity. This type of vaccine elicits stronger immune response than dead viruses or subunit viruses. In this type of vaccine, it is important to pick a viral vector that is truly safe to avoid immune response against the chosen viral vector that will make the vaccine less effective. Viral vector vaccine used in humans include those against the Ebola hemorrhagic fever. It is also used in most veterinary medicine. Currently, the University of Oxford and AstraZeneca, as well as the Johnson & Johnson's Janssen Pharmaceutical Company, independently develop viral vector vaccine against COVID-19. Nucleic acid vaccine uses RNA and DNA to teach our immune system to target key viral proteins. It is among the newest and most promising types of vaccine since it is easier and quicker to design. Vaccines developed by the American companies Moderna and Inovio uses RNA and DNA in their vaccines respectively. Toxoids are vaccines containing inactivated toxins directed at the toxins produced by a pathogen. Tetanus and diphtheria toxoids have long been part of standard childhood inoculation series. They require a series of injections for full immunity. It is then followed by boosters every 10 years. Before we proceed to the next part of our discussion, please answer this question using 1 to 3 sentences only. The question is, how important is a clinical trial in vaccine production? Please write your answer on your activity sheet. You may pause this video while answering, and once you're done, you may now proceed watching the next part of this video lesson. Historically, vaccines could be developed only by growing the pathogen in usefully large amount. The early successful viral vaccines were developed by animal cultivation. But currently, advancement in biotechnology paved the way to the development of new vaccines using recombinant DNA technology. In this technology, the antigen-producing gene is isolated from the viral source. In this case is our hepatitis V virus. A plasmid is also isolated from a bacterial source that will serve as the cloning vector. Both the antigen-producing gene and plasmid are cut by the same restriction enzyme that will produce fragments with sticky ends. Sticky ends facilitate the binding of the viral gene and the plasmid, forming a recombinant plasmid. The recombinant plasmid is then introduced to a yeast cell, forming a recombinant yeast cell. These yeast cells are then placed inside a fermentation tanks where they will multiply together with the antigen gene they are carrying. The antigens produced by these yeast cells are extracted and purified as a recombinant vaccine. To further your understanding about the use of recombinant technology for vaccine production, let us now have this activity number 5 entitled Filipino Pride. You're asked to research about the research project of the Filipino pre-scientist Father Nicanor Austriaco, a molecular biologist and a visiting professor for University of Santo Tomas. Write a 5-sentence paragraph about the impact of a locally produced oral vaccine against COVID-19. So to test yourself if you understand our lesson regarding immunity and vaccination, you're given the remaining time to answer the assessment on your activity sheet. Please follow the instruction. Read and answer each question carefully. Write the capital letter of your answer on the space provided before each number. Let us now have question number 1. What type of immunity is composed of structures and cells that serves as our body's first line of defense against infection? The choices are A. Acquired immunity, B. Adaptive immunity, C. Passive immunity, and D. Innate immunity. Question number 2. What type of vaccine uses a microbe that had been killed usually by formalin, phenol, or application of heat? The choices are A. Toxoids, B. Inactivated vaccines, C. Live attenuated vaccine, and D. Viral vector vaccine. Question number 3. What viral material is introduced by Jenner to a volunteer in their farm to determine the validity of the claims by his milkmaids? The choices are A. Smallpox, B. Chickenpox, C. Cowpox, and D. Monkeypox. Question number 4. What type of vaccine is developed by Moderna and Inovio which uses DNA or RNA molecules to teach the immune system to target key viral proteins? The choices are A. Subunit vaccine, B. Recombinant vaccine, C. Toxoids, and D. Nucleic acid vaccine. Question number 5. Which of the following is used by a human body to develop immunity through passive natural immunity? The choices are A. Milk and placenta, B. Vaccine, C. Antiserum, and D. Natural exposure. Question number 6. Which of the following companies are not currently creating an inactivated vaccine against COVID-19? The choices are A. Moderna, B. Sinopharm, C. Sinovac, and D. Gamaleya. Question number 7. What type of vaccine uses weakened microbes that can mimic an actual infection and provide a long-term effectiveness? The choices are A. Toxoids, B. Inactivated vaccines, C. Live attenuated vaccine, and D. Viral vector vaccine. Question number 8. What type of immunity is present when an individual is exposed to an antigen either naturally or artificially, and the response of the individual's immune system is the cause of immunity? The choices are A. Passive, B. Natural, C. Artificial, and D. Active. Question number 9. Aside from physical distancing and proper hygiene, which of the following will be our best way to combat COVID-19? The choices are A. Balanced diet, B. Vaccination, C. Enough sleep, and letter D. Vitamin C. And for our last question, aside from personal immunity against certain diseases, what is also the main goal of introducing vaccine in a certain population? The choices are A. Herd immunity, B. Direct transmission, C. Herd vaccination, and D. Direct inoculation. And this ends our lesson about human immunity and vaccination. I'm going to flash some of the links that you can browse on the internet if you want to further your study about this revolutionary procedure. Before we end this session, please be reminded to finish all the activities from the activity sheet. Once done, make sure to let your parents sign your output before submitting it next week. Again, this is your teacher facilitator in biotechnology, Mr. Marian Rodrigo D. Marinas III, reminding you to stay safe and healthy and see you soon.